Good evening, everyone. Welcome to another free session on Botany Insider's YouTube channel. Very good evening to all the students who have joined in. Right, we'll be learning about or we'll be revising cell biology in this particular session. Right, first we'll be talking about the most important topics from this particular unit, and then we'll be continuing forward with the revision of cell cycle and cancer both. Let's see how much we can cover up in this class because we have to understand and revise the topic also, and we'll be understanding and revising some of the PYQs also from the recent years. So stay tuned until the end of the video. You'll be learning a lot of things. Good evening, Mamta. Okay, thank you so much. I hope it was helpful for you, right? Okay, let's begin with the class now, right? So I've already taken up two of the sessions on Monday and Tuesday. Please make sure that if you want to uh, get a flow of your unit number one revision, you can watch that particular two classes. You can see the link for the same in the description box. That is for biochemistry. I took up two classes in which we learned about various small topics with questions and applying the whatever we have learned in the question then and there, so it will help you out. Let's start with cell biology. Before that, if any of you is targeting the June 2024 paper, then this particular we are having a full course going on for June 2024 batch in the minimal amount possible. It's going on our application. You can download our application available both on the Android as well as on the iOS. So if you wish to enroll in the course, we have a number of courses running for December batch also, for the next June batch also. So if any one of you who wish to enroll in the same, you can download the app and get yourself enrolled, right? You'll be getting hard copy notes in this full course. All the recordings, all the units, along with the part A, every single thing will be provided to you, right? Then for the June 2023, we have a mock test series going on with 100 plus test available in 599 only. So if any of you who is willing to like prepare with the question series, this particular thing would help you out. Let's begin with our class now and let's learn about the unit number 2, that is cell biologies. First, let's have a list of the most important topics. If any of you who has missed, I today uploaded a 32 short topics video on the YouTube channel. Any one of you who's willing to get through with these topics, you can, if you want a list for the same, I have uploaded a video for the same. You can download, like you can watch that particular video and make a list for the same. So from cell biology, very first and the foremost important topic is cell cycle. Every single year you can get a question from here, both from the topic about what cell cycle is. We have a number of concepts in the cell cycle. First thing is, what are the various phases in the cell cycle? I'll be revising the same in this particular class. Then the transition concept, that is, how does the transition from one of the phase, like for example you have two main phases, the interphase and the M phase, right? So in the interphase you have S1, G1, S, then G2, then M. In the M phase you have, this is your main mitotic phase, or depending on whether the cell is undergoing mitosis or meiosis, we'll be having differences in our phase over here. So let's revise about this in a bit. Then the next most important topic from which every year we are looking into a question is nucleosome concept. And that too, in nucleosome concept, majorly the question is from the model. There are two major models in nucleosome concept. First is your zigzag model and the second one is your solenoid model. From this particular thing, questions have been asked very regularly. In addition to that, the complete organization is again very important. How does the complete nucleosome organize and get arranged in the nucleosome? This is also important. In addition to that, the concept of transposons is also getting repeated over here. In last 2 to 3 years, we can easily see question from transposons from the various classes, basically or majorly from your examples of the various transposons that we have. So you can expect one question. I'll try to take up revision class on all of these segments. In addition to that, the concept of your various organelles that we have, you can get one or two question like it's not very common, but still you should have a very clear basic understanding of all those things. But from the transport of proteins, from the basics itself, when we learn about plasma membrane, you can get a question from the transport of proteins, which is a very very important and a bit lengthy topic as well. In this, the sodium potassium pump is of very very good importance. You can get a question from there as well. Okay, so keep a note of these topics. Do not skip at least the revision of these topics, because every single thing is important and you can get a question from these topics easily. Right now, let's revise the entire cell cycle in detail. I'll give you the gist of the entire cell cycle in the most detailed manner possible. So just stick by it. Let's understand. Right? Talking about what is cell cycle. Right? So what is cell cycle all about? Basically, cell cycle, it is a series of sequential events that is occurring for one cell to divide into its daughter cells, right? Based upon the cell that is undergoing division, whether it is a autosome or whether it's a sex cell, based upon the same, you'll be having a difference in the type of the division going on. Based upon whether it's an autosome or it's a sex cell, that is, it's a reproductive cell, you'll be having a difference in the same. Right? What is the difference in the two? So there are two type of divisions, right? What are the two type of divisions? The first type of division is your mitosis, and the second type of division is your meiosis. What is mitosis? What is meiosis? Understand this. Mitosis basically is you have a normal cell, an autosome undergoing mitosis, and this autosome gets converted or it gets divided into two of the daughter cells. This is mitosis. Over here, you have 2n as the ploidy level. In your daughter cells also, exactly similar 2n ploidy level will be there. In case of meiosis, you have initially, let's say, the parental cell, the mother cell. It will be getting divided into four of the daughter cells. Very important. Mitosis is equational division. This is equational division. Equal division is going on. Meiosis is reductional division. Why it is reductional? Because you are having your 2n in the mother, and in the next generation you have four of the daughter cells getting produced. All the four will be having half of the ploidy number. This is why it is meiosis. Why is it so? Because in meiosis we have the formation of gametes. Male gamete and the female gamete is getting formed, right? Male and female is getting formed. So when there is fertilization, when the fertilization will be going on, you'll see the ploidy level getting back to your 2n level. That's why in the gametes we require the ploidy level to be n. Otherwise, the maintenance of the ploidy in the generations would be impossible. That's why meiosis is there, or meiosis is seen in the sex cells or the reproductive cells, and in the normal cells that we have in our body, that is the autosomes. In that we see the mitosis going on. So remember the difference between the two. Now talking about the cell cycle. So cell cycle is what? It is a sequence of the events that is occurring for a specific cell to divide. Now division is of two types. When we look into the cell cycle, there are two phases. There are two main phases of cell cycle. Which are the two phases? One is your interphase and one is your M phase. Now, M phase could be mitotic phase or meiotic phase, depending upon which particular cell is it occurring in. It can be mitotic or meiotic. If it is mitotic, you'll be having four stages occurring. If it is meiotic, you'll be having you'll be having first meiosis 1 and then meiosis 2. In case of your interphase, you'll be having three phases, three of the sub phases going on: G1, S, and G2. G2 is the phase in which we have the main DNA replication occurring in. G1 is the main preparatory phase for your DNA replication to begin. G2 is the phase in which all the checkings are done, that whether do you have the normal DNA replication converted or done properly or not. In addition to that, we have the nuclear disintegration and all those things. We learn about that as well. In mitosis, you normally have prophase, metaphase, anaphase, and telophase. In this mitosis also, you can again divide it into two: cytokinesis and karyokinesis. One is cytokinesis and the other thing is karyokinesis. And if I want to write it down in correct order, I'll be writing karyokinesis on the top and then the cytokinesis. So this is karyokinesis going on, and then we'll be having cytokinesis going on. Karyokinesis is what? Karyokinesis is division or the segregation of the nuclear content. Cytokinesis is when you have the actual two cells getting segregated from each other and forming two daughter cells. In meiosis, you have meiosis 1 and meiosis 2. Meiosis 1 will have prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2 will be having prophase 2, metaphase 2, anaphase 2, and telophase 2, because you need to have the reduction in the number of the chromosomes. So, for that, second meiosis is also required. In this meiosis, in the prophase 1, you have this one being the longest phase of all time. Over here, you will be having five substages: leptotene, zygotene, acatin, diplotene, and diakinesis. Leptotene, zygotene, acatin, diplotene, and diakinesis. In your packet, we have the main event going on, which is known as your genetic recombination, which we call, or which occurs via the process of your crossing over. Why is it occurring? Crossing over majorly occurs so that you do not have any, like, for the recombination to occur, so that the meiotic cells can have recombined DNA. You can have varieties, you can have variants in the upcoming new generation. So, for diversity, for recombination, the Packetin stage have the genetic recombination going on with the help of crossing over mechanism. Then we have metaphase 1, anaphase 1, telophase 1. Then you have prophase 2, metaphase 2, anaphase 2, telophase 2. And after this particular stage, you'll be having your final division of the four cells, right? So, this is about your interface and the M phase basic information. In addition to that, there's one more important concept which is related to your transitions going on. So, transitions are what? Transitions are, because obviously your cell is undergoing, or your cell is getting proceeding from G1 to S, S to G2, G2 to M. So, what do you need? You need to have some kind of checkpoints. So, for checkpoints, you have CDKs and your cyclins working in properly, working in association with each other. So, there are various transition. Let's say G1, then S, G2. In M also, prophase, metaphase, anaphase, and telophase. So these changes are occurring at almost every stage. Over here, you have G1 to S transition going on. At S to G2, you have S to G2 transition going on. Over here, you have G2 to M transition going on. And the next important transition occurs here from metaphase to anaphase, which is known as metaphase to anaphase transition. In the first transition, we check that whether the cell has made up the MCM helicase or not. MCM helicase, it's the important enzyme that is required for the DNA replication to occur. In S2G2, it is checked whether the DNA has replicated properly or not. In G2 to M, all the things are cross verified again, that whether whatever machinery was required, whatever things were need to be done, is everything completed or not. In metaphase to anaphase, it is checked that whether our cell or our chromosome and the sister chromatids, are they connected to the spindle fibers, and are they connected to the, and is the kinetochores actually in the complete proper orientation, and do we have the equatorial plate in line or not? Because in the anaphase, your sister chromatids will be getting pulled apart from each other towards the opposite poles, and then you will be having your final stage going on for the final cell to divide into two. So every transition requires a specific cyclin, a specific CDK for it to work. Both cyclin and CDKs will be working in close association with each other so that they can work, because individually they cannot perform the functionality. CDK stands for cyclin dependent kinases. Kinases, so that means over here phosphorylation at specific positions are going on. Very, very important. Remember all the things, very important. From this slide itself, you can get a lot of question. So go through this. If you find any difficulty, you can ask. Otherwise, I'll be moving forward with the other segment. Go through this, everyone. It's a very good gist of the entire cell cycle. I've given you a good overview, and this will help you out. Try it, everyone. Then we'll be discussing about the G1 to S transition also. And one more thing, in metaphase to anaphase transition, what do you have in metaphase to anaphase transition? There is degradation of enzyme, the degradation of specific proteins going on. Activation of a protein like separase is done so that your sister chromatids can easily be separated from each other, because there's a protein known as cohesin which keeps the two sister chromatids in close association via or on the kinetochore side. So for removing that off, you need to have a separase enzyme activated. So in metaphase to anaphase, this separase enzyme gets activated via the degradation of securin, which is a protein again which keeps the separase in the inactive state. So securin gets degraded, separase gets activated, cohesin degraded, and finally you have your sister chromatids ready to move apart to the opposite poles. Check, everyone, whether things are clear to you or not. Good evening, Suresh. In addition to that, remember one more thing, metaphase is the main uh, phase in the entire cell cycle where we can see individual chromosome very clearly on the equatorial plate, right? So remember, metaphase is the phase where we can see individual chromosome, again important. Packaging is the stage where we have genetic recombination going on. Metaphase is the stage where you have the individual chromosome seen. uh, S is the phase, like synthetic phase or the synthesis phase is the stage where the DNA replication is going on. G1 is the phase where MCM helicase get synthesized. All these things are important. Okay. Understood, everyone, till here? Any doubt, any problem? Please ask. Otherwise, I'll start with the next thing. This will help you in the revision of the entire concept very effectively. You should have a very clear picture of all the topics in the same manner as I have uh, depicted cell cycle in similar pattern. Wherever processes are going on, wherever you have a mechanism going on, you should have a very clear picture of the entire thing. And if you have such good clarity of the concepts, no one can stop you from qualifying your examination. Any doubt, any problem? Right, let's move forward. Now, see over here, there was a question asked. See, the question is individual chromosomes are clearly seen in which of the phase of the cell cycle? This question is in was asked in June 2022. First option is G0, second is G1, third is S, fourth is M. So, answer for this particular question will be obviously option number 2, that is your metaphase. So, individual chromosomes can be seen in which particular phase of cell cycle? You can see it in the metaphase, that is your M phase. In this question, there's a bit of ambiguity because they have written down M phase. You can correlate it to the metaphase also, and you can consider it as the mitotic phase also. So, whichever is comfortable to you, you can correlate, but they should have clearly mentioned that whether they are talking about the metaphase or are they talking about the mitotic phase. So answer would have been option number D itself, any which way, right? So fourth is the correct answer. Simple question, basic question, right? In addition to that, let's continue with the next part. If this is clear, just let me know in the chat section. We have just discussed, see over here you have just seen that in the metaphase, what do we have? We have the individual chromosome C, and the same question was there in your June 2022 paper, right? Clear, everyone? Right, let's proceed. Now let's try to recall. See over here, you have the G1 phase, the S phase, then the G2 phase. Then in the G1 phase, cell growth is going on. In the S phase, you have the DNA replication. In the G2 phase, you have the preparation of the mitosis. Right? In the cell division, majorly we have prophase, metaphase, anaphase, and telophase. And finally, the cytokinesis. This is the main cell division going on. Right? Now, let's understand about the G1 to S transition in detail, right? So, G1 to S transition is what? When you have your cell going forward from G1, or when it has completed the functionality in the G1, it is entering into the S phase. So, the cell will be cross verifying that whether the required content or whether the required points are there in your completed in your cell or not, right? So you have two of the cyclins and the CDKs working in. First is your CDK4, 6 and cyclin D. Then you have the second one, that is CDK2 and cyclin E. So CDK4, 6, it will be causing the partial phosphorylation of your retinoblastoma, because retinoblastoma is required. Retinoblastoma acts as a negative inhibitor of E2F, which is required for the activation of the various transcription factors in the further steps. So E2F need to be segregated or separated from your retinoblastoma. If it gets separated from the retinoblastoma, how will it be doing it? How will we be doing it? There need to be phosphorylation, or there need to be any kind of mechanism which can remove this off. So for this, what will happen? You'll have the retinoblastoma partially phosphorylated initially with the help of your CDK4 and 6. Then after that, we'll be having the complete phosphorylation with the help of CDK2 and cyclin E, which will cause the complete phosphorylation. And in the complete phosphorylation, E2F will become free from the retinoblastoma. E2F will be then entering into the nucleus, where we can see the activation of transcription factors like FUS, JUN, CMYC, all these will get activated and they'll help in the formation of what? They'll help in the formation of cyclin A, ORIC, MCM, helicase, which help in the G12S transition, because MCM helicase is required in your S phase where the DNA replication need to work properly, right? So DNA replication will be initiated in the S phase, but the protein required for the same will be there in, or will be getting, what do you say, synthesized in your G1 phase, and this is checked with the help of your G1 to S transition, right? So remember about this, and based on this particular question, this particular information, the question was asked. See over here, question is the progression across the G1 to S boundary followed by the entry into the S phase is promoted by the activation of which of the following protein complexes. So you have CDK4 cyclin D, CDK2 cyclin E, then you have CDK4, 6 and cyclin, and then you have CDK4, 6 and cyclin D and E both. So question is asking you progression across the G1 to S boundary followed by the entry into the S phase is promoted via which of the complexes? It is talking about the second of the cyclin and the CDK. So it is cyclin D, CDK2 and cyclin E. So, what should be the correct answer? Cyclin 2, CDK2 and cyclin E. B should be the correct answer for this particular question. B is the correct answer for this question. Simple question. If you know about the entire transition, if you know about how does this transition proceed, these are from my notes, like my self-prepared notes itself. So if you know about how does the transition will be occurring, you can easily get to your correct answer. So CDK2 and cyclin E is the correct answer for this particular question. I hope it's clear to all, right? Simple question. It was a 2 marker that was asked in June 2022. So remember about it. Keep a note of all, like there are various other cyclin and CDKs as well, which you have to keep a note of. So remember them, keep a note of all of them. They'll be helping you in the main examination because you can easily get questions from this segment also. Is it clear to all? Yes or no? Please let me know in the chat sections whether the concept is clear to each one of you or not. We have the next concept of cancer topic. So in the cancer topic, I'll take up this cancer topic revision tomorrow, and then we'll be proceeding forward with other revision segments as well, right? So I hope things are clear. And if any of you is targeting the June 2024 batch or the June 2024 examination, so we have a full course going on. You can enroll in the same in the minimal costing possible. And this is about the mock test series, which is going on for your examination of this upcoming examination for December 2023 with 100 plus tests, and it is available for 599 only, in which you have full length test also, in which you have uh, subject test also, general aptitude question tests also. So it will help you out, right? I hope things are clear, and I hope this particular session helped you out. Be there in the class tomorrow also. We'll be having our classes till December examination every single day. Okay, so all the best. Good luck. Take care, everyone. Prepare well for the examination. Also, I have uploaded a 32 short topic list. Use that, it will help you. Bye, everyone. Take care.